Can we start?
Yes, yeah. I'll sit in.
Do you want to moderate?
Yes, I can do. Yeah. So,
Thank you.
so hi everybody. Welcome to the first talk of the day. And yeah, if you'd like to go ahead.
Yep, I'll just share my screen then.
Is this too loud? Does this work? Okay.
Ask if the online judges here.
Wait, can you guys hear? Can everyone hear? Okay, it works. Okay. Hello, judges, and thank you for taking the time to evaluate our project this year. We're a team, GEMS Taiwan, and for our project, Fusarium World, we focus on the world's most famous yellow crescent-shaped fruit, the banana, which is currently being threatened by a fungus named Fusarium oxysporum, Forma specialis cubans tropical race 4, which is kind of long, so we'll simplify it down to FOC. Now, for a little background information, what we know as bananas are actually different groups of genetically identical bananas called cultivars. And this lack of diversity leaves them especially vulnerable to diseases such as FOC. One of these cultivars is actually the Cavendish, which accounts for over 99% of all exported bananas to the US and the EU. And this was actually caused by a similar pathogen called FOC race 1, which annihilated the previously popular Gros Michel cultivar in the 1950s, which forced farmers to switch to the Cavendish, which resisted the race 1. But now the same situation is happening again with FOC tropical race 4, and it's threatening to annihilate the bananas. FOC tropical race 4 has already spread from Asia to Australia to Africa, and more recently in 2021 to South America. And it's continuing to cause more and more damage in the world. And so in our first modeling effort, we predicted the areas at most risk of FOC invasion using Maxent, a popular open source software used by ecologists around the world. We found that the areas at most risk, which is also the areas with the most suitable bananas in the dark blue, found that the areas at most risk are also the areas with the highest suitability for bananas, which is in the dark blue. And in using these areas, we identified countries where we could apply our solution, such as Venezuela, Ecuador, Indonesia, and more. And furthermore, we simulated future suitable areas under climate change conditions in the next 40 years. Found that banana suitable areas will actually grow by over 500 million hectares, which is in the green, and most of the current areas will be retained, which is in the blue, and very little lost. This indicates that more bananas can be grown, but also the more damage that the fungus can cause. So what does FOC actually do to kill the plants? Well, FOC is a soil-borne disease and travels through the soil. And when it detects banana root exudates from the roots, it starts growing towards the roots and looking for any gaps, such as in stage 2 there, and travels into the vascular system and goes through the whole system, eventually reproducing and causing the signature yellow wilt of the Fusarium wilt disease. And for all of this, the solutions available, such as quarantine, fungicides, and even genetically modified bananas, don't solve the fundamental problem with FOC, which stems from the monoculture and the lack of genetic diversity. And this is where we present our solution, a fungicidal bacteria combating the fungus. And the hope is that our solution will slow and halt the further spread of the fungus, allowing more time for a more sustainable and genetically diverse selection of bananas for everyone to eat.
To more precisely approach the issue at hand, we analyzed the infection dynamic between FOC and the banana root. In the soil, the FOC chlamydis spores essentially germinate in response to the detection of the root exudates of the banana plant, and this root exudate contains malic acid. So by the principle of diffusion, the soil can be divided into malic acid sufficient zones, MA sufficient zones, and MA insufficient zones. For FOC to successfully attack and infect a banana plant, it must enter this MA sufficient zone, then penetrate the roots. As such, we can exploit this fact, the fact that FOC must extend its vulnerable hyphae into the MA sufficiency zone, and we devised a theory solution of using a chitinase secreting engineered rhizobacteria to combat FOC through the destruction of the invasive hyphae. By allowing the bacteria to proliferate in this MA zone, the bacteria will form a protective layer over the root system, and inhibit FOC and hence stop fusarium wilt. However, the downside of using general effectors such as chitinase is that the effect of our bacteria cannot be specified to FOC itself, and therefore unintended environmental consequences may occur. To address this and mitigate the consequences, we devised a toxin-antitoxin kill switch system to achieve biocontainment. In order to curb the unintended consequences of our chitinase secretion system, we devised a malate-driven system to utilize malate as the permissive factor to achieve biocontainment around the roots. In theory, this will allow us to limit the proliferation of our bacteria in the malate sufficiency zone as shown in this diagram. So it would just be a layer on the roots. Moving on, to achieve the extracellular secretion of chitinase and actually attack the FOC to inhibit its activity, we utilize secretion tags in conjunction with our protein of interest, H8. Our chosen chitinase, H8, is a novel chitinase demonstrated in literature to possess inhibitory effects against ethoxysporum. And since we exclusively use E. coli during experimentation, we had to ensure that this secretion can actually go through the gram-negative cell wall of E. coli. So we used YEBF tag and fused it to the coding sequence of H8 using the GS linker in order to signal for the extracellular secretion of E. coli. T7 promoter was used to ensure a high level of constitutive production of the H8 such that the bacteria can combat the fungus at all times. For the transcriptional regulation of our kill switch system, we used MLER, which is a transcriptional activator responding to L-malate found in the MLE operon. Essentially, what it does in our system is allow the CCDA, which is governed under the PMLES, basically the promoter region and regulon controlled by MLER, to be inducibly expressed under the presence of malate, and thus allowing us to control the concentration of antitoxin according to the malic acid concentration. This is in contrast to the low and weak constitutive expression of CCDB, which is the toxin. The low constitutive production of the toxin will allow for the slow death of the bacteria outside of our permissive conditions, which is basically the malate sufficiency zones around the root. And therefore, biocontainment is achieved. In order to model the complex behavior of this construct system, we devised a kinetics-based mathematical model to ensure that the behavior of the constructs generally behave as we wish it to. And such, we had to first perform an exponential regression to estimate the malic acid concentration in the soil around the banana root, basically the concentration of malic acid in the root exudate, and we found that value to be roughly 24.3. And according to the results of our toxin-antitoxin model, it's shown that CCDA dominates over CCDB around 1 to 10 micromolar of malic acid, and this basically sort of corresponds to the fact that there are 24.3 micromolar of malic acid in the root system of banana. So this sort of demonstrates that our kill switch system can in fact contain the bacteria into our designated areas. For proof of concept, we mainly wish to ensure that the extracellular secretion of chitinase is actually achieved. And we will be doing this using two SDS page analyses. So we will be analyzing the intracellular fraction to ensure the actual production of the protein. And then we'll be doing a SDS page of the extracellular fraction to make sure that it's actually secreted. In order to extract the protein, we had to resort to a less conventional method due to equipment limitations, but the results are as follows. In the intracellular chitinase production half, there is a distinct 46 kDa band found in all trials involving Chi-AT and J on top. And for the bottom extracellular secretion trials, there are also 46 kDh bands found on the SDS gel, and this demonstrates that a portion of the chitinase has in fact been secreted into the extracellular space. And it can be seen that only a portion has due to the dimmer bands. For our other proteins, we successfully produced MLER, which is shown through the 34 kDa bands on the left, and also on the 34 kDa bands on the right for CCDA MLER. However, we were unable to confirm the production of CCDA due to the fact that the buffer system we used, lamellae, is unable to resolve small protein bands. For CCB, we sort of confirmed protein expression through confirmation of a protein band around 13 kDa. And also, again, we successfully produced chitin shades due to our confirmation of bands around 46 kDa. And to demonstrate the effectiveness of this secreted chitinase against fungus, we decided to simulate its effects against fungus by doing a co-culture assay against yeast. However, we were unable to get conclusive results due to the fact that yeast propagates too slowly. To improve this, we might use actual Fusarium oxysporum safer strains in order to perform experimentation, or we would simply just close in the distance of the interacting parties in order to get more conclusive results.
To understand the practicality of our proposed solution and its potential flaws, we approached experts who showed us the many nuances of the real-world implications of our project. Specifically, we reached out to Dr. Andre Drenth from the University of Queensland, who has an extensive research background in plant pathology, horticulture, and forestry. Dr. Drenth also published multiple works that characterize FOC. Upon discussion, he recommended the implementation of our engineered bacteria in early stages of plant life to overcome root turnover and ensure sufficient bacterial coverage of the root system. Reflecting upon Dr. Drenth's feedback, we plan to periodically apply our solution through the banana plant's entire lifespan to ensure proper bacterial concentration for defense. To this end, we devised delivery of the bacteria using a liquid inoculant. Our visit to the Taiwan Banana Research Institute, or TBRI for short, then expanded our previous knowledge of bananas and helped clarify Dr. Drenth's advice. By observing the plantlet production process firsthand, we saw the possibility of applying the liquid inoculant to the individual plantlet jars cultivated in plantation nurseries. To tackle the issue of covering the banana plant's developing root system, we devised to integrate the inoculant with existing drip irrigation systems through a Venturi pump. With an established implementation plan, we contacted Dr. Yu Liu, who informed us of the regulations surrounding genetically modified organisms. This inspired us to research further on both international and national policies regarding our proposed implementation and allowed us to create our entrepreneurship page. Our interview with her also helped us formulate our hardware into a more cohesive and applicable plan.
Then, to estimate the total market loss caused by FOC and the total saving our hardware can provide, we created two economic models. And the total market loss is estimated to be $15.3 million for a 30-year investigation timeframe. However, under full prototype protection, the economic loss is reduced by $16.7 million, thereby significantly buffering the crisis. In an effort to gain insight into public opinion, our team conducted a public opinion survey. To our surprise, many of our respondents are not informed of the ongoing banana crisis. Addressing the results of our survey, we plan to educate the public with three distinct approaches: presentations, podcasts, and infographics. We engage the public in traditional teachings and more detailed information. To create easy and digestible content for anyone interested in learning about biotechnology, we created a podcast detailing topics regarding to the ethics and the morals of synthetic biology and the new advancements in the scientific communities. In addition, our team created the infographic translation project, partnering with UI Oslo, Norway, Thailand RAS, and Korea HS to translate educational infographics. Through this, we hope to surpass language barriers and spread the knowledge of synthetic biology through entertaining as well as understandable formats.
So the dilemma posed by the rampaging Fusarium wilt. Our response is firm and composed. To prevent and mitigate future damage, there will be, Fusarium won't. Thank you for your...
So yeah, thank you very much for a really interesting presentation. I'm just going to encourage the judges to ask some questions.
Yep.
Um, um, I'll just go first.
Yeah, go for it, Amos.
Yeah. So initially in your data, when you're trying to verify the expression, I did notice that you're using SDS page gels, but I could not like tell if in respect to the controls or where the controls were. But also, like, have you attempted any Western blots for this kind of an assay?
Is there? I could hear the first part of your question, but could you repeat the second part of your question?
Oh, I'm just curious if you attempted any Western blots on this particular assay.
Oh, we didn't perform any Western blot because we didn't really have the equipment to perform Western blot, and plus the gel were sort of poorly made. Well, not poorly made, but excessive protein was loaded into the gels, which sort of resulted in there being quite blurry bands. And in response to the lack of control, essentially, we neglected to include control during the actual experimentation, but we sort of mitigated that by comparing to other gels that we did, basically cross-referencing gels and basically noting if there was a significant band on other similar gels performed at the same time.
Yeah,
We
that
simulated.
yeah, I'm just wondering what if there's any data that shows that you like besides this gel that shows that expression took place and that you have the protein available.
Is that a question?
Yeah, I'm just asking. Yeah.
These are the results. Yeah.
Okay.
Essentially this. Yeah.
All right, thank you. And I do appreciate the way that you formulated the BioBreak to create a containment strategy. And I really enjoyed the fact that you created a kinetic model as well for the tracking. So thank you for showing that, I guess.
I have one question. Can you hear me?
Yes, we can hear you.
Okay, so I want to know that what's your future plan? I mean, because it seems that you just performed the SDS page. I mean, what's the future experiment plan are you going to do to prove your concept and your data?
So for our future proof of concept plan would obviously be sort of to backtrack on our co-culture and more conclusively prove that our secreted chitinase actually has effect against fungus. And we'll try to contact bioresource agencies to source more biosafe strains of ethoxysporum to perform experiments with. In addition, we would like to also, of course, perform Western blot to more conclusively confirm the expression of our proteins, but also we would like to more experimentally determine the parameters in our kinetics model in order to better model the actual behavior of our toxin antitoxin system. And by basically doing this, we will be able to modify the construct designs that we used. And this will allow us to fine-tune the constructs such that biocontainment can be better achieved, because obviously the regulations around genetically modified organism, especially if applied to food, is very tight. So we would like to fine-tune our kill switch system to be much more precise to the value of malic acid concentration in soil. Yeah.
And of course, you need to try to purify the proteins and for the functional acid.
Yep. We
Very
added
nice.
the purification tags on the proteins, but unfortunately, we didn't have the time nor the resource to perform the purification. We'd obviously wish to in the future.
Yeah. Thank you.
So just to jump in on that, you can also use those tags as part of the Western blot to get a good idea of whether you've expressed that protein, which would be really, really cool to see.
Yeah, we plan to use antigens to do the Western blot.
Yeah, it's a really good way of showing it, especially if you've got quite a bit of extra stuff going on within that sample. So yeah, really good.
Hi, thanks for your presentation. You did a great job. Can you discuss more about the releasing of modified organisms to the soil? And also with they had plasmids inside, and these plasmids have resistance to antibiotics. And what would be the advantage, for example, against it only use the enzyme alone or immobilize the enzyme? Yeah.
So basically, if we are able to use a live bacteria, the live bacteria is going to reproduce, and then they're going to continue to produce this chitinase that we will wanted to fight Fusarium oxysporum. And if we only use maybe a source of chitinase in our soil, maybe we would have to add this chitinase maybe like chitinase solution, maybe like weekly, or maybe like a few days again and then again. However, if we use our designed bacteria, then we're able to maybe the bacteria would able to like stay in the soil and continue to reproduce and produce chitinase for a longer period of time. So that's the one of the advantages that we have for using a live bacteria.
So also additionally, using a live bacteria also basically allows for some sort of autonomy on this sort of biocontrol method. So basically, this bacteria will naturally adapt to the evolving root system of the banana plant. And as we mentioned in Dr. Drunt's interview, basically, he did notify us of this root turnover, and therefore by using a live bacteria, it's better able to adapt to this living system as a living system itself.
So just to go back to what I think was the first part of your question was, are you worried at all about those bacteria possessing antibiotic resistance and then sitting within that soil system? And have you thought about any ways in which you could get round that?
Do you mean if like the plasmid could be accidentally transferred into wild bacteria?
Yes, yeah. So yeah, I think that's possibly one of the worries, that if you have a plasmid with an antibiotic resistant system, that could be transferred to other bacteria.
So my answer would be that because I think what the toxin is in our plasmid. So the toxin is actually in our plasmid, and it is the CCDB that we mentioned in our previous presentation. So basically, if the plasmid is transferred into maybe like other bacteria, then they would also contain the toxin that is constructed in our plasmid, right? So basically, if so our way of preventing our bacteria to like not like maybe leaving the banana plantation is that when our bacteria leaves the banana plantation, then they won't have enough malic acid to produce CCDA, which is the antitoxin of CCDB. So I think there's no such worries for maybe other bacteria to maybe adapt our plasmid, because like the overall idea is that when they leave the plantation, then the toxin would be produced, and then it will eventually kill the bacteria by binding with DNA gyrase, and then preventing from transcription to occur. Yeah. So.
But maybe with the legislation, you have to check that part.
Yeah, you might just have to check that part with legislation, depending on the way you're deploying it and stuff like that. Which, it was really interesting to see that you'd spoken to people about not just the legislation where you are, but around the world. That was really good that you engaged thinking about the wider aspects of it. I thought that was really good and a really good job. So that's probably that's all we've got time for. But just to thank you again for a wonderful presentation and a great project as well. Thank you very much. So I think.
Do I just press leave?
Yes, yeah, I think we've got all the other judges now.
Should we take a picture?
Yes, yeah. You have to just do it really quickly because we have to move between rooms. So
can we take a quick picture? I guess we could maybe stand here and then behind the screen.
Oh no, go on. We'll let you do it. Honestly, it's fine. I'm gonna nip out as you've got a full set. But yeah, that's all right. That's all right because there'll be another room that